champions, I welcome you all to Score 360 out of 360 in NEET Bio within 250-day series, and today we are going ahead with the third topic of our chapter, Principles of Inheritance and Variation, and this is all about the dihybrid cross. As of now, we have completed monohybrid cross and things like that. We have to upgrade ourselves, right? So yes. Now what did we cover in the previous session, children? We spoke about incomplete dominance that was shown by Snapdragon. Then we spoke about codominance, which was shown by our blood groups, and then pleiotropy, which was about the seed shape as well as the star synthesis in the case of pea plants. Okay, we saw all these things, we solved certain questions as well, and I gave you one homework. Well, before getting into the homework, let's see how many number of questions were asked in the previous year. It was 6 number of questions, so 6 into 4, 24 marks in total. This is a very high weightage topic. We cannot miss this. Okay, now going ahead, this is the homework that I gave you, children. Let's see who all got it correct. ABO blood group is controlled by gene I. So it is saying gene I is the one that controls ABO blood group, both A, B and O. Now, which has 3 alleles? Okay, A, B and O would be having 3 alleles and shows codominance. There are 6 genotypes. How many phenotypes in all are possible? So basically they are saying genotypes can be 6, as in if they are saying IA gene is an allele, IB gene is another allele, and IO gene is another allele. Now they come in different combinations for giving out the blood groups. Okay, and these combination, for example IA and IB came, so I have IA, IB. So this blood group will be AB. Since I have both the alleles, and this is what you mean by codominance, both of them are dominant. Now if IA and IO came, then it would be A blood group because A is dominant over O. Or if IB and IO came, then B is dominant over O, again you would get B blood group. Okay. Now for O blood group, you need both the alleles as IO, then you get O blood group. About this we studied previously. They are asking you how many phenotypes are possible. Genotypes can be many. Here only we have discussed about 4. 2 of them extra are there. Okay, I will tell the 2 extra things also. So, okay, there is, if father and mother both are giving IA, it will be A. Father and mother both are giving IB, it will be B. There are 6 genotypes possible. They are asking you what is a possible phenotype. So I see either you can have B blood group, either you can have A blood group, either you can have O blood group. Same thing is repeated above there as well. Agree? Same thing is repeated over there. So the 3 possibilities are there. Only 3 phenotypes are there, children. Do we have an option like that, 3? Yes, we have option like that. Phenotype is basically the feature that is exhibited, that is shown, the physical character. So basically this is something physical that can be shown. Okay, when I do a blood test, it comes to my knowledge that I have A blood group. No one will be able to see what allele am I carrying. Genotype is something different. Okay, so it is 3 in number. Cool. Now let's go ahead with today's topic, that is dihybrid cross. So before starting reading the NCERT, I thought that I should be explaining this to you in a whole manner. So let's begin that. Until now, children, we were speaking about monohybrid cross. Monohybrid cross. What do you mean by monohybrid? As in, when I took a pure breeding tall plant and a pure breeding dwarf plant, when I crossed them, what did I get? I got capital T small t, again capital T small t, again capital T small t, and capital T small t. Okay, I call this as a hybrid. I call this as a hybrid. This particular generation is hybrid generation. Why? Because now they are tall, but they are tall like this. It's not tall like this. They are not homozygotes, children. These are homozygous, whereas this is what? Heterozygous. This is what, children? Heterozygous. Now, hybrid I call when there are 2 alleles coming together, 2 dissimilar alleles which are coming together. At that point of time, I call it heterozygous. Okay. So please understand, both of them, they are pure breeding, they are not hybrids. The generation that is coming ahead of them which has both the gametes, both the different alleles in them, okay, not both the gametes, both the different alleles in them. This is one allele, this is one allele for height. So this is what you call as hybrid. Now over here there is only one hybrid, which is why I call it mono hybrid. This is the hybrid thing that is happening. Okay, now let's go and talk about dihybrids. The scenario is going to change. Let's talk about dihybrid. So over here I have to take two characters initially. So just like how you are taking this, I'll be taking round seeds of pea plant, okay, and then there is wrinkled seeds of pea plant. This is a character. This is a character. Basically it's about the shape. It's about the shape. These are the different alleles. Similarly over here we were talking about height. These were the different allele group, and both of them were homozygous. Over here also they were homozygous. But I'm saying I'm putting one more thing over here, that is plants having round and yellow seeds, okay, pea plants having round and green seeds, okay. So I'm writing the codes also. Capital R is for round, small r is for wrinkled, okay. Now capital Y is for yellow and small y is for green. Cool. Now you have to imagine this just like how I am telling you. So imagine that there is one chromosome over here, there is another chromosome over here. Similarly you have one chromosome over here, one chromosome over here. We know, right, chromosomes basically come in pairs and each pair will be having one allele. Either it can be same allele or it can be different allele. So over here one chromosome is having R, other chromosome is also having R. Here one chromosome is having small r, other chromosome is having small r, okay. Now going ahead, over here in this chromosome you have capital Y, you have capital Y, that is the genes for yellow coloration. Over here you have an allele for small y and another allele for small y, okay. This is their parent's chromosome. Now you know, among a pair of chromosome, only one chromosome can be going ahead into the gamete, because from the parents to the gametes, if you want to form, you have to halve the number of chromosomes. Remember in human beings 23, usually we have 46 number of chromosomes, okay, all of them are paired. We send one pair, one side of it. So basically halving of 46 will be 23, 23 we get from father, 23 we get from mother. Now we have 46 number of chromosomes. Similarly in plants also things work like that, okay, things work like that. Over here the number of chromosomes are halved, one can come from each pair, okay. So from here and here, if I take one, you tell me, if I take this or this, anyways the gamete will be having capital R itself. Agree? Do you agree or not? The gamete will be having capital R itself if I take this or this. What about this? From here in this particular pair, if I take one, similarly it will be just capital Y. So can I say that all the gametes that will be produced by this plant will be having capital R, capital Y, okay, chromosome, agree? Similarly over here also, if I take this or this, small r, if I take this or this, similarly it will be small y. So all the gametes that will be produced by this particular plant will be small r, small y, agree? Done. This is law of segregation, and law of segregation does not have any exceptions. So law of dominance has an exception that you saw, that is incomplete dominance, and was co-dominance and stuff like that, right? But when you talk about law of segregation, it does not have any loopholes, okay, or any exceptions. I'll show you the law of dominance, law of segregation as well. So according to the law of segregation, they are saying though the parents contain two alleles during gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. It cannot receive both of them, either one will be gotten. Now, if there is homozygous parent, just like how do we have as of now, capital R, it produces all the gametes that are similar, while a heterozygous one produces two kinds of gametes, about which we have not studied it yet. We will be talking about heterozygotes as well. As of now, we are talking about homozygous condition, where it's capital R, capital Y. All the gametes will be similar over here, and from one pair of allele only one goes. This is law of segregation. Please remember it. Okay. Now, so this is done. This is done. Now, if I try to fuse this, okay. Now, if I try to fuse them, I get next combination of chromosome. One is this, and the other one I am making like this. Okay, fine. Or I should be making in equal manner. Wait, just a second. So I got one chromosome, another chromosome, and then I got one chromosome, another chromosome. Okay, so over here I got the one that was having R from father. I got the one that was having small r from the mother. Similarly, I got capital Y from the father, and I got small y from the mother. Okay, that is a new combination of pairs that we got, 23, 23, 46. That is why it looks different, because we get both of their character, right? Now, over here, you say, what will be the phenotype of the plant? What will be the phenotype of the plant? How will it look? Phenotype as in, as I always say, physical feature. You're talking about the physical feature. How does it look? So, capital R is there and small r is there. Capital R will dominate over small r. Yeah, and capital R stands for round, children. So, can I write round here? Yes. Okay. Similarly, over here, capital Y dominates over small y. Okay, and capital Y is, children, yellow. So, all the plants that you see over here, they will be round and yellow. Now what is the genotype if asked? What is the genotype of F1 generation? This is F1 generation, right? What will be the genotype of F1 generation? Capital R, small r, capital Y, small y, and still they are round and yellow. Same thing we got here, right? All of them were tall, but they were heterozygotes. They were not homozygous just as we got, they were heterozygotes. Over here also, if you see, all of them basically are round and yellow, that is the dominant feature, but all of them, they are heterozygotes. Now, over here if you see, they have two alleles, two dissimilar alleles. They also have two dissimilar alleles for different trait. So, this is for one trait or one character, this is for another character. So, can I call this as a dihybrid? If this was monohybrid, because it was talking about just one character and it was talking about the two alleles of a character, it was monohybrid. Okay, when it was coming together, you were calling it hybrid, but this is only one character, so you called it monohybrid. Okay, now over here you see there are two characters and both of them, they are heterozygotes. So, can I call them dihybrids? Yes, I can call them dihybrids. So, parents are not dihybrids over here, they are pure breeding. The product that you get in F1 generation, that is dihybrid, children. Okay, that is dihybrid. Okay, there's an important thing which I wanted to say, which is why I am mentioning very much about this thing. Okay, so phenotype of all the F1 plants are round and yellow, and the genotype is capital R, small r, capital Y, small y. Okay, now Mendel was like, we have to anyways, as I said, we have to do self-pollination, then only I can come to a conclusion, just like how I did over here. Okay, it was also self-pollinated, and the ratio that we got after self-pollination was 1:2:1. This is genotypic ratio, and 3:1 is the phenotypic ratio. All were tall except one. Okay, so this was the thing. Let's see what happens over here. So please remember, capital R, small r, capital Y, small y. We are going ahead. Just a second, children, it's too cold. Just a second. Yeah, over here. So I have capital R, small r, capital Y, small y. Here also I have capital R, small r, capital Y, small y. Now, for imagination, I am making the chromosomes as well. This is one chromosome, this is another chromosome, this is one chromosome, this is another chromosome. Over here, you will have capital R, here you will have small r, over here you will have capital Y, over here you will have small y. Okay, same thing we can draw it there also. So 1, okay, wait. Over here we have capital R, over here we have small r, over here we have capital Y, over here we have small y. Now during gamete formation, what will be the gametes? What are the gametes going to look like? So for this particular and for this particular thing, what are the gametes that we could find? Let's see. So from this particular pair, as I said, one allele could come down. It can be capital R first. Okay. From this particular pair, one can come down, and this is capital Y, suppose. So there is one gamete like this. Is that the only gamete that they can form? No, children. Next, capital R again can be there, but with capital R the small y can come. So this is the next gamete that we have. Okay. Now what? Small r is there. Small r can come with capital Y, and small r can come with small y. Agree? So we have 4 gametes again. Over here also, if we see, so just a second, children. Can I make it little, let's say, shorter? Wait. Capital R, small capital Y, capital R, small y, small r, capital Y, and small r, small y. Okay. Now over here also same thing happens, same parent, no? So capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. We have 4 type of gametes. 4 type of gametes, children. Please remember this. Okay. Now I'll tell you one trick where you can find the number of gametes that will be formed always. So check for the number of hybrids. Here you have 2 type of hybrids, 2 hybrids, dihybrid. So 2 power n. Here n should be the number of hybrids. So here 2 hybrids are there. So I will replace n with 2. Now 2 square, what is 2 square? It is equals to 4. So basically I will get 4 gametes. Now if I have a scenario like this, sorry to interrupt, but yes, it will be really helpful for you. Now if I have a scenario like this, AA, BB and CC. Okay. And you are asked, tell the number of gametes. What will you do? Check for the number of heterozygotes. Okay. Check for the number of hybrids. 1, 2, 3. This is trihybrid. So what do? 2, 3. 2 by the power 3. So 2 into 2 into 2, what is it? Basically 8. You will be having 8 gametes, children. But you have to look for the hybrids. Now if the condition was like this, AA, BB and CC, what is the number of gametes that will be produced? Tell me. There is only one hybrid over here, right? So 2 is to the power 1. Basically, you just have 2 gametes. Baki rest, everything will be the same. Okay. Okay? Got it? So one gamete will be having capital A, B, C. Other will be having small a, B, C. These are the 2 gametes that will be formed. So I hope you understand this. So we have to know the number of, why did I say that knowing hybrids as well as the number of hybrids is very much important, because this questions can come out as the number of gametes that is being formed. So over here you saw 4 type of gametes are being formed. 4 type of gametes are being formed. Now what, ma'am? How can we see that these are the gametes? Now we have to find the whole F2 generation. How can we do that? So here comes the role of Punnett square. So let's make Punnett square. I don't know if it will fit over here, but we will try our level best. Okay. So we have to make 1, 2, 3 and 4. Similarly, 1, 2, 3 and 4. Okay. Now let's write capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Who was the person who got the Punnett square? If you remember, can you tell me? If you remember, can you tell me? It was in the comment section, you can tell me. Reginald C. Punnett, right? So I'm writing over here. Okay, these are the 4 gametes. Now this side, same thing. The other parent will also give the same gametes. So what do we have? Small r, small y. Okay, please do not mug this up. It will be really, when I was a student, I used to mug this up because my teacher was not able to explain. Okay, so I was like really terrified of it. I was not able to understand, so I mugged it up. But I did very big mistakes in the day of my exam, on the day of my exam as well. So I don't want you to repeat the mistakes. Okay, please remember the number of gametes. Think in the format of this. It will be really easy for you. Only one chromosome for a particular allele can be coming down. So either it's R or R. Either it's Y or Y, small y. Okay, think like that, and you have 4 possibilities of the gametes. Now 4 possibilities of gamete, let's see the possibilities of the offspring that will be produced. So capital R, capital Y, capital Y. Agree? Again, capital R, capital Y, small y. Okay? Capital R, small r, capital Y. Similarly, capital R, small r, capital Y, small y. Okay, if you see, R is dominant over this, so it will be round. This is also dominant, it will be yellow. All of them, if you see, they will be round and yellow. We got 4 of them are round and yellow. Going ahead, again capital R, capital Y, small y, capital R, small y. Okay, we got round and green. Okay, one round and green. I'll change the color. Wait, this is an exception, so we have to change the color. Oh, why 2 of them? This is capital R, small y. Okay, fine. Next thing, what do we have? Capital R, small r, capital Y, small y. Agree? Done. Going ahead, we have capital R, small r, and capital Y, small y. Okay, 2 of them we got. They are both round. And both are green in color as well. Okay, round and green. Going ahead, we have. So I'll stop changing the color because it will be time utilizing. But still, capital R, small r, capital Y. Okay, capital R, small r, capital Y, small y. Yes. Small r. Okay, there is a change. Let's make the change. Did it go? No. Now over here, there is small r, capital Y. So the first time we saw a wrinkled and green one. Sorry, wrinkled and yellow one. So there's a new combination. We never saw wrinkled and yellow. Wrinkled and green was a combo. Wrinkled and yellow was not a combo. Okay. So here also we saw a new variety that was round and green. That is also a new combo. We were always studying about round and yellow. Now going ahead, what do we have? Small r, capital Y, small y. Okay, same thing, a new combo that we got. Going ahead, capital R, small r, okay, small y. Again, round and yellow, just like them. Okay, now going ahead, capital R, small r. Oh, this is capital R, small y. Okay, this is again the same type. So wherever the variations are there, I'll just make a white. That will be better. Otherwise, our whole time will be going. Okay. Now going ahead, what do we have, children? Small r, capital Y, small y. So we have got wrinkled and yellow for the first time. So this is again a new combination that we have got. Okay. And then last but not the least, we have small r, small y. So it is wrinkled and yellow. Agree? This was wrinkled and yellow. This was wrinkled and yellow. This is now wrinkled and green, the original variety. Okay. Now we are going to see for the genotypes, because I know it's very much confusing. Not the genotype, the phenotype. Okay. First, let's see the phenotype. So I see 1, 2, 3, 4, 5, 6, 7, 8. Wait. Did I mess something up? 1, 2, 3, 4, 5, 6, 7, 8, 9. We have 9 of them which are round and yellow. Okay, and I am telling you we would be having wrinkled and green as well, because that is what we started with. This is wrinkled and green. Now you will be having two different combo. So just make a combination of it. Initially it was round and yellow. So put round with green now. You will be finding round with green. Let's see who all are round and green. Okay. So I see this is round and green. Okay. This is also round and green. And then this is also round and green. I have 3 of them which is round and green. Then I can make the new combo, that is wrinkled. Wrinkled was always green. So now let's make it yellow. Wrinkled and yellow. Let's see. Small r, and big Y. So small r, big Y. Smaller, big Y. Smaller, big Y. How many of them? 3 of them. So basically this is the phenotype. You got round yellow, you got round green, you got wrinkled yellow, you got wrinkled green. Is there something that you have to mug up? No, children. Please remember, the initial variety was round and yellow and the other one was wrinkled and green. You cannot obviously make round and wrinkled seed, no, because this is the same character. Round and wrinkled cannot come together. Either it can be round or it can be wrinkled, but the color can change. So you can pair this with this, okay, or you can pair this with this. So round yellow or round green. Similarly, wrinkled yellow or wrinkled green. So the most common one will be the one that is most dominant. So we started with a dominant parent that was round and yellow. So all of them, 9 of them will be round and yellow. Okay, 9 of them will be round and yellow. This is the phenotypic ratio that I am talking about now. Okay, now going ahead, talking about round and green. Okay, before keeping there, wrinkled and green is there, which is our recessive parent. We started off with a recessive parent over here, remember. So wrinkled and green also will be there. I am keeping it 1. So the one that was recessive, it is the least there. The one that was dominant, it is the most there. It is 9 and that is 1. Now in middle you have to fill 3 and 3, that's all. So round and green will be 3, equal. All the different varieties that came in are equal, and the other one will also be 3. This is what, children? This is the phenotypic ratio. This is the phenotypic ratio of F2 generation. Now again, you need to find the genotypic ratio. They would not be asking about the whole genotypic ratio, but you know how to make if they are asking. Okay, I mean, I'm not saying they won't ask the ratio. I'm saying they won't ask you to make all the genotypes. Okay, all the different genotypes. Mostly it is not asked. So listen to me very carefully. Genotypic ratio you can remember like this: 1:2:1, and then again 1:2:1, and in between just put 2:4:2. That is a double of this. Double of this will be in the middle. So 1:2:1, 2:4:2, and then 1:2:1. This is the genotypic ratio. Okay, that is the number of genotypes. You will be finding only one small y over here. Okay, similar to that, you will be finding just 2 of them over here, like this and this are the same. Nowhere else you can find it. So you have different genotypes over here. That is what is mentioned over here as well, children. Okay, so phenotype is this. Phenotypic ratio is this. This is the most easiest thing. Genotypic ratio is this. Okay. Now, if you see just round and the wrinkled part, okay, if you see just the round and the wrinkled part, you will come to a conclusion that almost this is round 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 of them are round. 12 of them are round. Let's talk about the wrinkled one, okay? Wrinkled. 1, 2. Okay, wait. 1, 2, 3, and 4 are wrinkled. 4 of them are wrinkled. Now, if you cut down, I can get 3:1 ratio, just like how I was getting in monohybrid cross. In monohybrid cross also, I got something like this only. Remember? Remember when you crossed a monohybrid cross, it was something like this. Wait, just a second, I'll show you. So, you had capital T, small t, you had capital T, small t, you crossed them, okay, and then you found that one was like this, the other one was like this, the other one was like this, and the other one was like this. So 3 of them were tall and 1 was dwarf children. Similarly over here also in F2 generation, this is about F2 generation, okay. Here 3 of them are round, okay, another one is 4 of them are wrinkled, fine. We are not saying what round, it can be heterozygote or homozygous round. Now if you are talking about the yellow and the green thing, same thing happens over here as well. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. And this one more thing I am not able to find. Anyways, 12, there will be 12 yellow and there will be 4 green in color, which is why 3:1 again. So this whole thing is a cumulative expression of 3:1. So 3:1 basically round and wrinkled, 3:1 basically yellow and green. Okay. Now if you are not able to understand that, I can make that also. So please remember, this is given in your NCERT actually. So they follow this thing. Over here we have round and this is wrinkled, okay. Now this is yellow and this is green children. This is yellow and this is green. Now 3 into 3, 9. So we have 9 round yellow, okay. 3 into 1, we have 3 round green. Now 1 into 3, so we have 3 wrinkled yellow, and then 1 into 1, we have 1 wrinkled green. So 9:3:3:1 ratio, phenotypic ratio you can get by this equation. Now when we are talking about monohybrid cross, I said that day, but it was not that much clear. So it was shown like this. Wait, AX and BY whole square. So over here I am saying A is half capital T, B is half again, but it is small t, and this whole equation is a square plus b square plus 2ab, right? So I can make like that. Now if I do a square half t into half t, it's basically 1/4 capital TT, okay, 1/4 capital TT. Going ahead, half t into half t, you get 1/4 small t small t. Now 2ab, 2 into AB, okay, what do you get? 1/2 will be, right, this was the equation, was it the equation? Yes. So you got the whole thing like this. So 1/4 will be capital T, 1/4 will be what children? Small t, and another half will be capital T, small t. Okay, got it? Okay. Now this was the thing, and I hope that we are finished with the dihybrid cross as well. Please check for the number of gametes. Okay, please check for the number of gametes. If they are asking like this, what is the number of gametes? How can it say? There is only one hybrid over here. This is not hybrid. So what will you say, children? It will be 2:1. That is, there will be only 2 type of gametes that will be formed. It can be either R capital Y, or it can be capital R small y. That's the same thing. Okay. So this is about the dihybrid cross, and with this information I want to tell you about the third law of genetics, which is law of independent assortment. What do you mean by law of independent assortment? Basically, law of segregation was saying that in a pair of alleles, only one allele will be going to one gamete. So this allele went to this gamete, this allele went to this gamete. Only one allele can go in a gamete. Okay, that was law of segregation. Now law of independent assortment says, when you are talking about a pair of alleles, that is, this is a pair, this is a pair, one pair of allele will be moving down to the gamete independently of another pair of allele. So capital R didn't say that I just want capital Y with me, otherwise I won't be going down and forming a gamete. It's not like that. It formed capital R, capital Y, but over here you saw that capital R was going with small y as well. So this pair of allele is independently assorting from this pair of allele. That is what you mean by law of independent assortment. Okay. Now, if it's too difficult for you to understand this, once more I can give you an example and see. Okay. So, you have a friend Joe and you have another friend Rach. Okay. Then you have a friend Nisha and then you have a friend Alf. Okay. So, over here Joe, which is capital R, Rach, which is small r, Nisha, which is capital Y, and Alf, which is small y. Okay. From this both thing, one person can go and join the competition for race. Only one. Capital R can go. Now from this particular team, only one person can go. Either it can be Nisha or can be Alf. So I'm saying this. So the Y will be only going. Similarly, this person can also come for this match, right? This person can also come for the match, that is small r. If Rach decides to come, it's small, yeah, it's small r. Okay. Now, again, if Nisha wants to come, it will be capital Y. Anyone among them can come, anyone among them can also come down. It is independent. You cannot say that Joe will always go with Nisha or Joe will go with Alf. You cannot do that. Okay, you cannot do that. So basically they are independently assorting from each other. I hope it's clear to you. Fine. Now, going ahead, we will be just reading our NCERT and see if you are missing down anything. Okay. So, Mendel also worked with and crossed pea plants that differed in two characters as seen in the cross between a pea plant that has seeds with yellow color and round shape and the one seeds that has green color and wrinkled shape. So, we are talking about two characters just as we spoke. Mendel found that the seeds resulting from the crossing of the parents had yellow colored and round shaped seeds. So, round and yellow was dominant trait. Here, can you tell which of the character in the pair yellow green and round wrinkled shape was dominant? So, you know round was dominant and yellow was dominant because all the progeny in the F1 generation were round and yellow. Thus, yellow was dominant over green and round shape dominant over wrinkled. These results were identical to that he got when he made separate monohybrid crosses. For example, he just took round and wrinkled one and then he fused, then also he got the same result that round was dominant over this and yellow was dominant over green plant. Okay. So, yes, let us use the genotypic symbols Y for dominant yellow and small y for recessive green color seed, capital R for round shape and small r for wrinkled seed shape. The genotype of the parents can be written as capital R, capital Y and small r, small y, small y. We already did it over here. Okay, fine. Now, going ahead, okay, the gametes capital R, capital Y and small r, small y unite on fertilization to produce the F1 hybrid, that is your capital R, small r and capital Y, small y. Okay, that is exactly what is written over here. They are also saying the same thing, that basically this is segregated into 3:1 ratio. Over here, in yellow color plants also it will be yellow and green color plants also it will be segregating into 3:1 ratio. Same thing that we already studied over here when I was talking about this. Okay. Now, what is something new for us? This is something new for us. You saw the Punnett square. I already made it for you. If you do not remember, you can think like this also. This is a more clear version of it. Okay. So, there is only possibility of capital R coming down, capital Y coming down. Similarly, small r coming down, small y coming down. Both of them if fused, you will get this. Now, from this particular capital R, small r, either one of can come. So, one combination can be capital R with capital Y, other can be capital R with small y, and the same thing over here. So, you have to make the Punnett square and then you have to practice, you will get better understanding of it. Now, this is an information that we should not be missing, we should be reading it once. Where is it? Yes. Okay, in dihybrid cross, the phenotypes round yellow, wrinkled yellow and all this thing appeared in the ratio 9:3:3:1. Such a ratio was observed for several pairs of character that Mendel studied. Okay, not just this. The ratio of 9:3:1 can be derived as a combination or series of this thing, series of 3 yellow, 1 green with round yellow, 1 wrinkle, the one that I drew already to you and I showed you, right? Now the derivation can be written as follows. So you fuse this with this, you fuse this with this, and you get whole thing. We made it. Now the Punnett square can be effectively used to understand the independent segregation of the two pairs of genes during meiosis and the production of eggs and pollen. Basically they have eggs and pollen instead of sperm and gametes, right? In the sperm and egg. So egg and pollen will be having either of the gametes and then both will be fusing. Now 50% of the gametes have gene R. Another 50% will be having small r. Now besides each gamete having either capital R or small r, it should also have the allele capital Y or small y, because you're talking about round and yellow or round and wrinkled, right? The important thing to remember here is that segregation of 50% of capital R and 50% of small r is independent from the segregation of 50% of capital Y and 50% of small y. So capital R, small r, capital Y, small y. There are 50% chance for this to come, 50% chance for this to come, 50% of chance for this to come, 50% of chance for this to come. So this is independent of this thing. Both are very much independent. Okay. The same thing I have written over here. What is something extra that, let's see that. Okay. Thus there are 4 genotypes of the gametes. The 4 types are capital R, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y, each with a frequency of 25% or 1/4 of the total gametes produced. So if you're asked this question, children, what is the percentage of chances that this gamete can come? So it is 4 gametes are there, so it will be 1/4, that is 25%. 25% of chance for this particular gamete to come if the cross is dihybrid like this. If the cross is dihybrid like this, then 1/4 of chance is that this particular gamete will be coming down or this particular gamete will be coming down. It's 25% each. Please remember it, write it somewhere. Okay, when you're talking about a dihybrid cross. Now, when Mendel self-hybridized the F1 plant, he found that 3/4 of the F2 plant had yellow. The same thing that we covered, there is nothing extra. One line I deleted from here so that I can put it in the next slide, that is about the law of independent assortment. So when 2 pairs of traits are combined in a hybrid, segregation of one pair of character is independent of other pair of character. So we know that the one for shape was segregating independently from the one for color. It had nothing to do with this. This is law of independent assortment. Do not get confused with law of independent assortment and law of segregation. Law of segregation says from a pair of allele. Law of segregation is for a pair of allele. That from these guys, either capital R can go, either small r can go. But when you talk about law of independent assortment, it's about 2 pairs of alleles for different character. So it is saying this assorts independently of what is there over here. Either it can go with this, either it can go with this. Okay, please understand the difference. And law of dominance, you know that. So basically we have completed law of dominance in the previous classes, law of segregation as well, and law of independent assortment. Now law of dominance has exceptions. It has exceptions. Co-dominance, we have incomplete dominance and pleiotropy and all of them. Segregation, no exceptions. No exceptions. Okay. Now, law of independent assortment, exception is there, and that is what we are going to study in the next class. And the exception is linkage, because linkage definitely occurs. It's not that always you will be having 2 separate chromosomes for each allele. So it can be on 1 chromosome also. That's a problem, and you will come to know about that as well really soon when we talk about linkage. Okay. So law of independent assortment has exceptions, and law of dominance also has exceptions. Yes. Now children, my dear champions, can you read this question and can we try to solve this? Tell me the answer before I try to solve this. In Mendel's experiment with garden pea, round shape, he has written like that, okay, round seed shape was dominant over wrinkled seed shape. Yellow cotyledons were dominant over green cotyledons. Okay, you have to check this. Sometimes they can give the opposite also. We can say that green is dominant according to our studies. So in that case, you have to be sure that what is capital Y representing. So if it is yellow over here, it is yellow. It's all good. Now, what are the expected phenotypes in F2 generation of the cross? They are asking you, if this is the cross, okay, what is the phenotype of F2 generation, not F1. So in F1 we got that all of them were capital R small r, capital Y small I, all of them were round and yellow. They are asking about F2, we studied it right now, 9:3:3:1 ratio. 9 was what, children? Round and yellow, okay. This was round and green, this was wrinkled and yellow, and this was wrinkled and green. The minority will be the recessive one, the majority will be the dominant one, rest combinations are in the middle. Now let's see. Round seeds with yellow cotyledons, and wrinkled seeds with yellow cotyledons. That is one thing, okay, that is there. Next, only round seeds with green cotyledons. Only term does not come. You cannot have only cut, because you have 4 different things that comes in, and here only is not mentioned, which is why this is the correct answer. This came in 2006, children. Okay, I hope you got the answer. Tell me who all got it correct. Now going ahead, due to the cross between capital T, okay, and small t, small r, the resultant progeny shows what percentage of tall and red. So we are given the phenotype. That we have to produce tall and red colored plant. Now this is tall, this is red, this is not tall, this is dwarf, and this is again probably white or something other color. Okay, now listen to me very carefully. They are asking about tall and red percentage. How will you do that? Will you cross all these things? Will you keep on crossing all these things? 1, 1, 1 each of them? No, we should not have to do that. So over here you know that this is homozygous, this is also homozygous. So when homozygous, this and this pairs, every time it will be capital T, small t that you get. So all of them will be tall. Agree? All of them will be tall. Going ahead with this and this, when you talk about this and this, okay, now that if you want you can cross, okay, here, 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 here. So over here if you cross this, capital R small r, capital R small r, small r small r, small r small r. So 50% of them, okay, if you see 50% of them will be capital R small r. And all of them, they are tall. So I can say the one that has tall and red, obviously it will be 50%. You have to talk about red also, right? So tall and red combination will be 50%, children, over here. Okay, so for this you don't need to calculate the whole thing, do not need to make all the things. Just understand this is homozygous, all of them, if they cross, whatever they do, one capital T will be there with the small t. So this all of them will be tall for sure. You have to find the red one. So for that you can do this. You found that 2 of them were red, 2 of them were not. So 50% of them were red. And you're asked about tall and red. You were not asked about how many tall were there. If you're asked about how many tall, then 100% of them are tall. You're talking about tall and red. So it is just 50%. So this is the correct answer, children. Got it, chitkuts? Over here, in a plant, red fruit is dominant over yellow fruit. Okay, in a plant, red fruit is dominant over yellow fruit and tallness is dominant over shortness. Okay, fine. If a plant with genotype capital R, capital T is crossed with a plant with small r, small t, you have to find what percentage of them will be tall and red. Okay. So again now, let's see for the homozygous one. This is homozygous, so you don't need to touch them. You know capital R will dominate over small r, so all of them will be red. Now if you talk about this and this, can we make that? Capital small t, what are the gametes? Capital T, small t, okay. When you fuse, half of them will be capital T, small t, half of them will be small t and small t. So basically 50% of them will be tall. All of them will be red, okay, but only 50% of them will be tall. So I can write 50% of them is tall with red fruit. Done? Easy? Now, in order to find out the different types of gametes produced by a pea plant having the genotype wait. Okay. In order to find out the different type of gametes produced by a genotype having AABB, okay, it should be crossed to a plant with the genotype what? In order to find out the different type of gametes produced by a pea plant having the genotype, okay, it should be crossed to a plant with the genotype what? What will be the genotype? That is the question. Try to solve this first. Okay, done children? Over here, understand, you are saying you have to find out a different type of gametes, okay, produced by a pea plant that is having this thing, and hence to find the different type of gametes, what should you be crossing it with? So if you cross it with something that is dominant all over, all the things would be different, all the gametes will be having just the dominant character. If you if all of them, if you see they have some dominant character, you won't be able to find things out. So for finding the gametes out, you need to always pair with a recessive parent. That is A, small a, small b. This is known as test cross. So test cross was initially done to find the genotypes of this thing. Over here, if you need to find the gametes, basically the gametes will be formed from this particular plant only. So they are basically trying to ask you the genotype itself in a way. Okay, so basically for that you need to perform a test cross. When you perform a test cross, you will be able to find the gametes that will be produced. Okay, this is the next question. So over here we solved this question, we solved this question, we solved this question, and we solved this question. This is your homework for the next thing. This I am not explaining it now. Okay, try by yourself and then we will come to them. Fine. And over here I will explain you the homework question which I gave you, because I am giving you homework as the other one. This is your homework question, please remember it. Okay, you have to do it and let me know the answer. This is the homework. Now over here, this question we can do by ourselves. So let's see. A human male produces sperms with genotypes AB, okay, AB, AB and AB, pertaining to two diallelic characters in equal proportion. What is the corresponding genotype of this particular person? If this particular person is giving these type of gametes, that means it will be definitely having capital A, it will be also having small a, that person can be having capital B, and that person can be also having small b, as you can see over here. So this will be the combo. So let's see which is correct. So this one is correct. This was an easy question, which is why I thought that I should be shifting the question. So this was your homework. I did your homework, but you have to do this. This is your homework now. Please try to do it. Okay. So that's all, children. I hope you were able to understand really well about dihybrid cross. Now, if you still have doubts, do let me know in the comment section. I will try to solve all your doubts. And one more thing, I hope you are having a really good time for preparation and everything, and I know that midterm is also going on, so all the very best for your midterm exams if you are in 12th standard. And for others, all of you, just be happy, just prepare and give mock tests regularly. I see that lots of people have not watched the mock test. Please go try, attempt the mock test and find the solutions for the same. Yeah, so bye bye children, take care, have a really good day. Do not forget to subscribe to our channel.